Guinevere, may we ask you to come up on the stage?
So from University of Groningen, Guinevere Lageveen-Kammeijer is joining us. She is an expert in coupling mass spectrometry to capillary electrophoresis, which is an exciting combination. And she's applying this work on glycomics, looking at sugar modifications of proteins, and actually applying this on the single cell level. So that's quite impressive. I'm looking forward to your presentation.
Thank you. So next to glycomics, I also would like to discuss a little bit about single cell proteomics. But maybe why are we doing single cell proteomics? Why do we want to do single cell at all? And that if we look at the oncology field, there's still a huge challenge that we need to overcome. Because if we look worldwide, we have 20 million new cancer cases per year. And with that, because most of these cancers are still diagnosed at a very late stage, treatment is often not very successful. So we also have high mortality rates with 10 million cases per year worldwide. So this really demonstrates the urgent need for new early stage biomarkers, but also therapeutic targets. And we believe that we need to dive deeper into the cell complexity, because if we look, for example, at a biopsy, what we can see by eye is the cell morphology. It's quite heterogeneous. We see that there are different populations. But if we look into these populations, we can not see the molecular heterogeneity by eye. So we need to dive deeper and investigate whether or not we have, for example, subpopulations within a certain population. And this is what we would like to discover with single-cell proteomics, but also single-cell glycomics. So the workflow that we have in our lab is that for now we use a cell suspension of 200 cells per microliter in PBS. And then with this cell suspension, what we're doing is to sort these cells using a cell-in-one system. So for those that are not really familiar with the system, I just have a small video to illustrate how this works. And you can do this in a 96 well plate. So what you can see over here is the piezo dispense capillary. And what we do over here is to apply acoustic waves. And with that, we generate very tiny droplets of 100 up to 500 picoliters. And we have a camera on the end of this capillary. And what you can see here are different zones. So this is actually the volume that will be dispensed in our well. And this is the so-called safety zone. So there we don't want to have any cells remaining. And then everything is monitored, and now you can see that we can set, we can set the diameters, the elongation, and everything is monitored using this system. And with that, we also, every time when there's a cell being dispensed in our well, a picture is taken, so we can always look back whether or not there's a single cell being captured. So after that, we can also do the cell lysis and protein digestion within the system. So everything is automated in a 96 well format. And after that, we use a nano-elute system, so an LC system coupled to mass spectrometry. And in our case, we're using a timsTOF SCP, so specifically dedicated for single-cell proteomics. And then we perform our data analysis. And actually, I would like to dive a little bit deeper into the data analysis because we have been struggling a little bit. What is the right method to analyze our single-cell proteomics data? And we have two different tools that we use for this. You can use either Spectronaut or Diane. And if you're familiar with these tools, you're maybe also familiar with the terms such as method evaluation, matching, as well as boosting. And the difference is that you treat your data differently. With method evaluation, you kind of look at your individual cell. If you do matching, you also combine it with the full cohort of your single cells. So you also combine, you kind of generate a library of all your cells and then have a look which of your proteins can be identified there. And if you do boosting, in that case, you have a bulk sample along with your cohort to really boost your library, to hopefully boost also your signals. So how does that look like if you, for example, look at HeLa cells? So if you here have Spectronaut, if you do individual matching, so this is the precursors, then if you do matching, you can really nicely see that we significantly boost our signal, but also using boosting. And how does that relate is that what we also notice that on average, it's a little bit lower if you do boosting compared to matching. And our hypothesis is there that it might be related to a less reliable FDR control in SpectraNow, because it's not made to have kind of an imbalance in your sample cohort. It's more made if you have bulk samples, that all your samples are bulk, and not just 1 or 3 samples that are higher amounts. And then if you look at our protein groups, you can nicely see that if we then look at the matching, it seems that we kind of lose the biological variety that we have with our individual samples. So it's more homogeneous. But if we then look at boosting, we kind of seem to kind of keep this variety compared to the individual, which might represent the biological variability better. And then if we then again look at the matching, it could be that we're masking this biological variety because we make our samples more homogeneous. We also applied this to a different type of cells, but we saw this exact same results. Again, we see with matching, we really have higher precursors IDs, and then with the boosting, it's a little bit lower, potentially to a less reliable false discovery rate. Then maybe dive a little bit deeper into this data, and if we look at the A549 cells, and we look at how many of our peptides are identified, you can see with the individual, we have a lot of single hits. And this is the percentage of the total protein groups per cell, and here you can see the absolute amount. And what you can see with matching is that we observe a higher percentage of peptides that we have around 5 up to 14 peptides per protein group being identified. And then if you then look at the protein coverage of the boosting, we also still have a really high hits for the singles. So potentially, again, illustrating this less reliable FDR rate. And then if we compare that to Diane, what we've noticed is that in general, we have lower hits. So we see less of our proteins and less precursors. To illustrate that, if we compare that to the SpectraNow, with the individuals analysis, we have plus 13% gain, almost double if we do matching, and 40% gain if we do boosting. And then the discussion kind of happens, or we're now exploring, what is actually correct here? So should we use SpectraNow or should we Diane? So which one is more reliable? Could it be that SpectraNow in general just finds better the precursors, or could it be that we have a stricter FDR control using Diane? And this is something that we're now currently further exploring, but what we've noticed also in relation to a previously, just recently published article is that also the group of Krijsveld kind of found out that Diane seems to be more reliable in matching compared to SpectraNow. And we kind of see the same thing if you look at the boosting analysis. So further research on this aspect, we will focus more on the false transfer. So what is really happening and how much of the data that we identify is actually a false discovery. So this is something that is ongoing. Then I also would like to highlight our workflow using laser capture microdissection. So also here, we use our timsTOF SCP system. And for those that are a little bit less familiar with laser capture microdissection, this is how it works. You have a tissue, and together with our pathologist, we cut out the specific regions of interest, and then we can analyze the proteome of that sample. And we were wondering, okay, how much sample do we really need? How much volume of our biopsy do we need? And in this case, we use SpectraNow for data analysis, and here you can see a quick summary that if we use, so this is again the precursors, this is the number of identified protein groups. So if we use a volume of 120,000, which is roughly equivalent to 24 cells, we can still identify around 3,000 protein groups. And you can nicely see that that doubles if you do the double amount of cells, but that we also reach a plateau and that we're not able to detect more than up to 4,500 protein groups. So it's not beneficial in this case to even go higher, and we were quite happy to see that with only approximately 24 cells that we can get good proteome coverage. If you want to know more about this, because further research, of course, is to really apply this to a clinical cohort, and we actually have one of our PhD students presenting a poster with some preliminary results of head and neck cancer patients. So feel free to look at her poster. So then switching to glycomics, because if we move back to this picture of the cell morphology and the molecular heterogeneity, is what we don't know a lot about is these sugars that are attached to our cells, to our lipids, to our proteins. And we really think that these sugars might be a key for finding new biomarkers. And why is that? Well, let's have a look at this endothelial cell. So this is the glycocalyx, so the extracellular surface of our cell. And all this fuzziness are glycans attached to our lipids and our proteins. And on top of that, if we use this analogy where we look at the spike protein, so here illustrated without any of its modifications, we're kind of looking at this bird without feathers. We have no clue how this bird is able to fly. But if we then start to incorporate its sugars, you can see it's completely decorated, dark blue with sugars. And now we can see how this bird is able to fly. And you can also probably imagine that if your proteins and your lipids and your cells are so heavily decorated with these sugars, it has a huge influence how they can interact, recognize, transfer. So we really need to study this more in depth. So the workflow that we have in our lab is focused on having our cell populations. We can use laser capture microdissection, but we can also use cell suspensions. And then in this case, what was already nicely announced at the beginning is that we use CE, so capillary electrophoresis, instead of LC. Why is that? Well, with the CE system, we have the advantage that we can get really good separation if you have charged or polar compounds. In addition, it has a really good separation efficiency, and we only need to use minimal sample amounts. And then we actually analyze our data using an in-house tool, Glycogenius. So what is the sample prep if we have our laser capture microdissected sample, and then it moves into the CE system? So first of all, we lyse our cells, and then we immobilize our proteins on a PVDF membrane plate. Then we add our enzyme called PNGase F to cleave the end glycans from our proteins and add an internal standard. Then we elute these end glycans from our plate, and then actually we label the glycans to add a permanent positive charge, which further enhances our separation with our CE system. Then it's time to analyze it on the CE system with the mass spec and then do the data analysis. And this is how such a profile can look like. So please bear with me that this is only from a 9 nanoliter injection where we used 1 microliter of plasma concentration at the start. And what you should also take into account is that actually we need around 5 up to 7 microliters in our vial before it can be injected. So we only inject a tiny, tiny fraction of that sample. Nonetheless, what we can nicely see is that we get a lot of different glycans that we can observe, and that with the CE we separate based on how many sialic acids, so these purple diamonds, are present on our glycans. So no sialic acids, 1, 2, 3, 4, and even up to 5. However, only injecting 9 nanoliters is very limited, and we want to go for single-cell glycomics. So we want to further push that, and might not sound a lot, but we were able to push that 10 times, so get an injection of 90 nanoliters, and still remain the isomeric separation that we need, because it's essential if we can distinguish the alpha-6 versus the alpha-3 linked sialic acids, because they have different biological implications. So with our optimized conditions, we were able to do this injection. And also just a small notification that we've managed to see that we have a limit of detection of an atomole level with our system. How does this look like if you, for example, then look at cells? So we use different protein amounts, or we kind of correlated our cell amounts to protein amounts. And then with our conventional injection, if you use high amounts, of course, you observe a lot of end glycans, so more than 200. But if you go lower, roughly 200 picograms, which is roughly equivalent to 1 cell, we were not able to detect any of our glycans. But then with our 10 times higher injection, we were still able to see the top 5 glycans. And we also applied it to, this was on a PA2S cell line, a cell line related to pancreatic cancer. We also applied it to the GY cells, and we got similar results. In addition, we also applied it to extracellular vesicles. And there we also could nicely see with 90 picograms, we could still see the top 4 end glycans. So our signal to noise with above 9, we can still see okay signals of our glycans. How does it look like if you look at the signal to noise of one specific glycan, so the most abundant one in our cells? Then you can nicely see this linear correlation. So this is with the normal injection, and then if we push it to 10 times higher, we really reach this limit of detection of 90 picograms of protein, roughly equivalent to a single cell. Moving back to the laser capture microdissection, we just received really preliminary results. How much cells do we actually need to get a proper glycomic profile of our tissues? So this was actually analyzed last week. So this is an illustration where we have roughly 1,000 cells as a starting amount. So that means that we have 7 microliters, around 1,000 cells in our vial, and we only inject 90 nanoliters of that, which is equivalent to 12 cells. So we kind of have a nice profile illustrating a lot of different end glycans. But I think what's also nicely to show is that if you look at our blank sample, we still observe some of these glycans, but way lesser amounts compared to our LMD sample. And we could still reliably detect 22 end glycans in our duplicates from this. So further research, we want to, of course, really go to the clinical samples and see, okay, how much can we see with these amounts? Can we even push it further? And hopefully next time I can show you some real clinical results of the head and neck cancer patients. So with that, I hope I could show you that we can do single-cell proteomics. However, it's really debatable what is the right data analysis approach there. So what are you aiming for? Also, what we're struggling with at the moment is how to normalize your data. And in the end, we also want to apply it for the LMD samples to the real clinical samples. Same for the glycomics. And also our timsTOF is equipped with an eye mobility modality. So ideally, we want to combine that together with our CE to even go further in the isomeric distinction of our glycans. So with that, I need to thank a lot of people that have been involved in here, especially Wenjun, Alinka, Hector, Kartika, Christopher, and Ying. And with that, thank you. And while we're going into Q&A, I also will show you a small video of our in-house software tool for Glycogenius. Thank you.
Thank you, Guinevere. Any questions for her? No? Overwhelming amount of work. I'm wondering how you would apply that and incorporate that with maybe metabolomics, from a metabolomics perspective. Mm-hmm. Yeah.
So yeah, with the glycomics, I think one of the things that we were struggling with is right software tools. You generate a lot of data, but still, for example, if you needed to look into your data, it took days to really do manual search. So hopefully with this tool, we can further speed that up and to also combine it in the end with the other omics layers that we have of the same samples.
And one question which always bothers me a little bit with single cell is, do you account for cell cycle at all?
No, not at the moment.
Yeah.
So that's indeed also something that we need to further dive into, because where you are in the cell cycle, probably you get a different proteome, glycome. So that is also something that should be further studied.
Yeah. Okay.
Yeah.
Thank you very much.
Thank you.